Welcome to Backstage at Upstage, a presentation of Upstage lung cancer, which uses the performing arts to raise awareness and funding for lung cancer research. Here's your host, the founder and president of Upstage Lung Cancer, Hildy Grossman.
Hi, I'm Hildy Grossman, and we're so excited to have you with us today backstage at Upstage. And here's my good pal, Jordan Rich.
Thank you, dear friend Hildy. Today, we're going to explore the Human Genome Project and the work being done right now with the study of genetics to help detect and diagnose lung and other cancers and to offer treatments and possibly even cures. Hildy, we welcome back to the podcast Dr. Jane Wilkinson, Senior Director of Broad Genomics Alliance Management of MIT and Harvard, and Assistant Biology Professor at the Koch Institute for Integrative Cancer Research at MIT, Dr. Francisco J. Sanchez-Rivera, who at this very moment is running a laboratory in Cambridge focusing on the human genome and its connection to cancer. Hildy, two outstanding guests. We can't wait to hear what they have to say.
Today, we'll try to make a complicated topic something even I can understand. Many of us have heard about the Human Genome Project, but most of us couldn't explain it to someone else to save their life. There are terms that are basic to the project, such as genes, genome, DNA, sequencing, and no, wait a minute, don't be frightened out there listening to this. We're going to make this all very simple and accessible for you. So I want to actually introduce someone to you who worked on the Human Genome Project, and she'll help us understand what a remarkable project this was. Jane Wilkinson, welcome.
Thank you, Hildy.
So first, could you explain some of these basic terms that we need to know to really understand what you're going to talk about in terms of your experience with the project?
Yeah, I think I'm going to start off by explaining what the human genome is and what our genomes are and what have they ever done for us. The human genome is really it's the biological map for everything that really goes on in our body. It drives a lot of development, a lot of diseases. It's good for some things, it's terrible for others. And I like to think about it as, it's a book. What we did was we basically created a book of the 30,000 genes that are in the human genome. That's about 3 billion bases, and they're divided amongst 23 chromosomes. So for me, the way to think about it is each chromosome is a chapter of the book. And all of the genes that are located within the individual chromosomes are really what drive a lot of the development within our body. And Francisco is gonna take the story from me later and tell us all about how these genes have led to us really discovering more about genetics, and especially cancer genetics.
So can I just jump in and see if I can understand this? I know the audience will too. So genes are things like they determine the color of our eyes and maybe how tall we are, and maybe if we have certain kind of talents, they're related to these genes. And so those are important particles inside of us. Is that right?
Absolutely, yeah. And the Human Genome Project actually started out with 1 or 2 humans. They were the contributors to the original Genome Project. I think what's really important for us to talk about is how far it's come when we try to understand the different genes and the different things that they drive. Is that original Genome Project was 1 or 2 people. We now have access to hundreds of thousands of human genomes, so we can really start to understand the differences between individual populations, and also very importantly, I think, especially for cancer, is the different populations as they exist within different environments. So it's incredibly important that we've taken the Genome Project into, now a library. If we use the book analogy, we came up with that original book, and now we've reprinted that book hundreds and thousands of times over in different populations and different parts of the world. So we really have a library of information now about genetics and genes and how they drive us and how they drive disease.
And so how did you get involved in this project in the first place before we go further in the science of it all? How did that happen?
Yeah, it was really an incredible journey for me. I was in the University of Liverpool, and I replied to this tiny little job advert in the back of a science publication. And yes, this was in the days of when jobs were advertised in print, and it was for a very small new institute called the Sanger Institute. So this was April 1993. I was actually looking to relocate from Liverpool, UK to Cambridge, UK. So this fitted perfectly with some life things that were going on for me. And what I did there is I joined an amazing person, Professor John Sulston, who actually went on to win a Nobel Prize and actually became knighted in the UK as well, to work with John on a project called the C. elegans genome. So the nematode genome. The nematode is a tiny little worm that lives in the ground. It has a very, very simple biological structure. It's what we call a model organism. It's something that we can interrogate very easily at a biological level. It regenerates quickly. It's really easy to visualize in a microscope. You can actually see how cells individually divide. So that was the first multicellular genome that was ever undertaken. Our success with the nematode, the C. elegans genome, led us to believe that we could take on this crazy project called the Human Genome Project. And what I like to think about with the Human Genome Project is really one of the greatest feats of exploration in human history. But instead of going to the planets or to the other side of the Earth, we were actually doing an inward journey of discovery, looking inside of ourselves. I think one of the things that's really important to point out is this really was the first truly international biology collaboration. It was an international team of researchers. The researchers came from 6 different countries, the US, the UK, France, Germany, Japan, and China. And it really was the first consortium-led big biology project that we've ever undertaken. It really defined collaboration. We started mapping the genome, so breaking it up into its chromosomes and into, some bite-sized chunks that we could all kind of literally wrap our heads around. We started mapping it in October 1990, and the genome was eventually finished in April 2003.
Can I ask you a question? So how does that happen? I mean, how does mapping happen? How do you break it up in chunks? What is this?
So back then, we used something called BACs, bacterial artificial chromosomes, and we mapped and assigned parts of the genomes to these artificial bacterial chromosomes, which really broke it up into bite-sized chunks for us. And by breaking it up into these bite-sized chunks, this allows us as individuals, all part of the consortium, to divide the genome up across the world. My particular division of it was I took on the chromosome 1 initiative. So I led the chromosome 1 sequencing component, and chromosome 1 is the largest chromosome, and that's my brag on the human genome. I led the largest one. But there were 2,800 researchers involved in this. And, it really was that first big, large-scale collaboration, but it also proved to us all that we could do this. We could take on really, really, really big problems. And as we come together as collaborators, we can really solve them. The genome was given an original budget of $3 billion. It actually came in under target, and we only spent $2.7 billion, which is a ridiculous amount of money when we think about genomics now, because right now we can do a person's whole genome probably for less than $500. And, instead of it taking 13 years, we can actually turn around an individual's human genome in a couple of days. So,
oh my God, isn't that incredible? Oh my God, this
is
mind-boggling, right?
It is to think how far we've come, but also, not how far have we come from a technology point of view. And there was a huge amount of technology development and leaps in innovation during the Genome Project itself to today. But really taking the data and understanding it at a biological level, that what we call translation, translating the individual genes into actually understanding how they define diseases and how they define development. It's been such a revolution of science.
It's amazing. So I have another question. So when you were assigned chromosome 1, right? That's because you're a star, you would get number 1. So how do they determine this is called chromosome 1? And what did you know about chromosome 1 to begin with?
Well, we've always understood the physical layout of the chromosomes. You can actually look at an individual's chromosomes from a drop of blood. We've always been able to see and understand the chromosomes themselves. You could also do things like chromosomal banding as well. So that can identify different, from an imaging point of view, we've always well understood and well characterized the chromosomes and what they've potentially been responsible for some very obvious diseases where the genetics are so very clear. What the Genome Project has allowed us to do is break that down at a word-by-word basis. So instead of just looking at the picture of the chromosome, we can now actually see everything that goes into it genetically on a base-by-base level. And Francisco, please jump in here. You're the university lecturer. You're the actual official teacher here. So I'd love to get your perspective too.
If you want me to put the importance of the Human Genome Project in the context of cancer, I'll be happy to do that. In doing so, I'll revisit some of the points that Jane did at the beginning, which I think are important to emphasize.
So I think before we jump into how we've done that translation, I want to pick up on a couple of large-scale projects that tumbled out of the Human Genome Project, which I think will segue really nicely into Francisco's work. There were a couple of projects that came out of understanding the genome at a basic level. We then created a project called the HapMap Project, and what that allowed us to do is understand genetic variation between individuals and different populations. ENCODE, which helps us to define the functional elements that come out of DNA sequencing. But the thing I really want to talk about is, and the project that I was involved in as we progressed, and as I came across the pond and joined the Broad Institute across the street from the Koch Institute, and they are a great collaborator of ours, is the Cancer Genome Atlas Project. So we affectionately call this TCGA. What we did here is we took 33 different cancer types, including 10 rare cancers. We did DNA sequencing on all of those cancer tumors from 11,000 patients, and really started to interrogate the cancers at a genetic level to really try and start to understand any genetic signatures that could be potentially shared amongst the different cancer types. It allowed us to really define different tumors and different cancers at a genetic level, really start to break them down at a molecular level. But one of the projects that, I'm sure we're all incredibly interested in and we've all thought about is a project called the NCI Lung Map. So this is the National Cancer Institute. So this is the US's federally funded and run cancer program. And what they've done is that they've looked at multiple different lung cancer types and are really trying to understand at a genetic level the changes in the different tumors, so we can really start to understand the tumor itself at a molecular level, but also start to offer very, very personalized and very predictive treatment plans based on the molecular and the genetic understanding of those lung cancer types. So that's been a huge success for the NCI. So Francisco, I'm going to let you kind of take it away from there. I've kind of maybe laid the tarmac on the road to cancer genomics and where we are today. So I'll let you drive the car.
Thank you, Jane. So maybe actually what I'll first do is revisit a few important points that you already made, because I think that even though we live in an era where we talk about genes and we talk about mutant genes being involved in diseases, I don't think that the most people out there fully grasp what this actually means. I constantly feel like we should be incredibly amazed at the fact that we can do all these things these days. So first of all, when Jane mentioned that we have sequenced many different genomes across the world, one important point to keep in mind is that these are all genomes composed of the same 4 letters. So all of our genes are composed of combination of just 4 DNA letters, which are A, T, G, and C. And the precise nature and order in which these letters are arranged essentially determine who we are. And specific strings of these letters are what make genes and other regulatory elements that Jane mentioned function. And so this is really important yes.
No, I was just gonna, I'm so sorry to interrupt you, but
No
again, I'm gonna stop you when I stop understanding. So, so what does this mean? There are these ADDC letters. Like, what does that mean? Like somebody typed on a typewriter and those are those letters? I mean, what does this mean? I don't understand.
It almost means exactly that, but A, T, G, and C are called DNA bases or nucleotides. These are the only 4 bases that are composing our genome. And so
They're
in
a chemical?
the order,
Is it a chemical?
they are chemical bases. Yeah. They're just called DNA bases, but they are essentially a chemical structure, an organic molecule.
So they're different chemicals that just get the title of 1 letter. Is that right?
Yeah, and they connect between each other. And actually the order and the way in which they connect is what determines, genes, and genes therefore determine traits, how we look, etc., but also, influence disease outcomes. And this is particularly important in the context of cancer. So maybe I can just use this as a segue to talk about cancer. So the reason the projects like the Human Genome Project initially and efforts like the Cancer Genome Atlas are so critical for understanding cancer is that cancer is largely a genetic disease. And that means that cancer is caused by alterations to our genes, which are made of DNA. So as I mentioned, all of our genes are composed of these different letters, and changes in the order of these letters are commonly referred to as mutations. And some mutations can cause cancer. They can also cause other diseases, but they can also influence the way we look. The different populations, different people have different sequences of genes, even though all 4 of us here have the same 20 to 30,000 genes, the sequences between our genes are slightly different in important ways.
This was really so clear now, because you hear the word mutation and, all you can think of is, what is it, the mutant turtles, or what does, what does that mean? But to just spell it down, like just the alteration of the sequence of those letters, that's amazing. It's great. Thank you.
Yeah, so not every mutation is bad, and that's something to keep in mind, because mutations actually fuel evolution. So organisms undergo natural selection over time through mutation, and that influences how they look, how they function, and how they acquire disease, and whether they have lower or higher risk for certain diseases. So not every mutation causes disease, and this is really important to keep in mind. So in the context of my own lab, for instance, we're interested in learning which mutations cause cancer and understanding why and how the cancer causing potential of these mutations can vary depending on the individual. So by that, I mean some mutations in some people can cause cancer, and the same mutations in other people do not cause cancer. And that's a very important point. Sorry, I thought you were gonna.
No, but I think that, again, this is super interesting in a general sense, because it feels like it fits with kind of more macro level of disease and problems. Like there are people who have herniated discs who can hardly move, and somebody else has the same herniated disc who doesn't seem to have a difficulty. And so why our bodies respond to the same, now you're at a micro level, but why our bodies respond differently is just, I haven't a clue, but it's really interesting.
Yeah, and it has a largely genetic component. And the only way we can understand these different genetic components and how they vary depending on the individual and across populations is through DNA sequencing or technologies that arise from DNA sequencing. Yes.
Where are we, if I can just jump in with only one question that matters on my end, and thank you, Hildy, for asking all the questions that I already had. But where are we on the time scale in terms of advancement? Because Jane laid out a beautiful chronology, and we know how technology is rapidly advancing as it moves through year after year, decade after decade. So if we were to put a scale up, say 1 to 10, obviously 10 is probably impossible for any scale, but where are we? Are we at a 3 or 4? Is that easy to quantify in terms of success and understanding?
It's a little hard to quantify in that way, but let me just actually put it in context. So today, when a patient goes into the oncology clinic, they undergo a biopsy of their tumor or a blood isolation if it's some leukemia or heme malignancy. Those cancer cells are sequenced. So the DNA of those cancer cells is sequenced. And some places can sequence the whole genome, but we don't really need the whole genome to carry out diagnostic and treatment decisions. But the point is that every patient goes and has their tumor or tumors sequenced. That is hugely important, and it still blows my mind when I think about it because it's routine. And the reason it's routine, and many of these DNA sequencing technologies are now routine, is because of the Human Genome Project. These technologies would have taken decades longer if it wasn't for the Human Genome Project international effort. So the reason why it's so informative to sequence these genes is that, again, mutations in these genes are what causes cancer. So essentially, we have within our bodies the seeds for cancer development. Because if we acquire certain mutations in certain genes, we will develop cancer. It's not some exogenous force that causes cancer. It is, to some extent, that causes mutations, but it's really the mutations in our genes that lead to cancer development. So by knowing exactly which genes in patient tumors are mutated, we can start making treatment decisions and diagnostic decisions. And this is hugely important. This is now routine in the clinic. So if you were to ask me, where are we in terms of 1 to 10, I would say we are about 8, because we're still trying to understand what all of these genes and what all of these mutations are doing. And this is actually what my lab is particularly interested in. So, we as humans have roughly the same genes, but again, not every mutation causes cancer in every human being. And on top of that, those specific mutations can influence the types of treatments that patients will undergo. So if a patient has a certain mutation, we know for some of them that they will be sensitive to certain therapies, but for others, we actually don't know whether they'll be sensitive or resistant. And actually, there are mutations that respond very well to certain therapies and actually respond very poorly to other therapies. And actually, some therapies can accelerate the disease progression if the drug to mutation combination is not correct. And again, this is so important to keep in mind because we are now able to connect genetic information to therapies.
We'll get back to the podcast in just a moment. As thousands of audience members know, Upstage lung cancer events, the concerts, are fun, meaningful, inspiring, and memorable. And you should know that we invest in cutting-edge diagnostic research to find lung cancer early and greatly improve on the 5-year survival rate. We also bring voice to the fact that young people get lung cancer. They really do. Unfortunately, doctors don't know how or why. Proceeds from our concerts support research to help find answers to these questions. Hildy Grossman and her team aim to entertain and inform, because the show must go on. Find out how you can help at upstagelungcancer.org. Now back to the podcast. Here's Hildy.
You're talking about, in terms of doing these sequencing of the genes from the tumor, from the biopsies, this is what leads to biomarker testing. Is that correct?
Yep,
Right.
exactly. Yeah.
And so what's going on in your lab at this point in terms of using this diagnostically? As you said before, you're trying to look at, well, and I think that's what we've certainly had podcasts on, not only biomarker testing, but targeted therapies, choosing a therapy to aim specifically at that mutation. What do you think needs doing in terms of diagnosing cancer, lung cancer early?
Yeah, so the diagnostics are very urgently needed, I would say. So for some cancers, most cancers, if they're caught early, you might have success in terms of treating that cancer or actually just removing the tumor, depending on the type of disease. But some cancers, for instance, pancreatic cancer, just to give an example, by the time it's found, it's often very lethal. So being able to diagnose that disease very early would dramatically shape and transform the way that cancers are detected or treated. Maybe some patients will not need to get treated, and maybe they're caught early, they would not suffer from metastatic disease, which is typically what kills cancer patients. So
Right.
one key
Metastatic
technology,
disease, meaning that the cancer will travel to other parts of the body.
Yeah. So there's a couple of things that come to mind. The first one is that genetic profiling is hugely important, because you could, for instance, genetically profile people that don't have cancer so that you can identify who's at a higher risk of developing cancer. There are certain types of alleles, which are versions of genes. Each gene that we have is composed of 2 alleles, one that we got from one of our parents and the other one that we got from the other parent. So those can be different. And sometimes you can inherit an allele that has a specific alteration, a different order of these DNA bases, or a different base at a specific place where it shouldn't be. And that mutation can increase the risk of someone developing cancer. So that's one way where I think we could potentially intercept cancer earlier, or even prevent it by knowing which alterations people have.
And so when you're saying genetic profiling, what are you talking about? Is that the whole gene map of the whole person, just to see what's going on everywhere? Or are you just talking about if there's something in the family that people are concerned about, such as lung cancer? That's another one, like pancreatic cancer, that does not have great early diagnostic tools yet.
Both of them are correct. Actually, you nailed both of them. And even though we are now getting used to technologies and things like 23andMe, they're actually the same principle. Knowing what types of single nucleotide polymorphisms, without getting too technical, this means that there are different letters in different genes depending on the person. So some of those single nucleotide polymorphisms can influence whether someone will get disease or not. And by doing this kind of genetic testing, you can capture some of those and have a better understanding of the risk that someone might have to develop a certain disease like cancer.
And how
So
is
that's
that
one.
how do they do that test? How would that be done?
This is as easy as isolating blood from someone.
So do you look at circulating DNA or?
So that's what I was gonna get on the second option. You're very advanced.
Sorry, I didn't mean to jump the gun.
you know a lot about some of the scientific jargon here, because the second one that I was gonna mention is the ability to through sampling blood isolate both circulating tumor cells, often called CTCs, or circulating tumor DNA, also called ctDNA, which is present on either by itself, actually in some cases, but also within the CTCs. And so often you can identify, well, in some cases, you can identify CTCs in people where you don't readily identify a primary tumor. So in theory, by sampling the blood of someone, you can ask whether a person has CTCs or ctDNA. And if they do, you can actually sequence the genes that are present in that cell or the DNA that is circulating in. And you can actually use this to detect specific mutations that might influence cancer development or progression. So for instance, yeah.
No, I was just wondering where your lab is in terms of looking into these things now.
Yeah, so the key point here is that every cancer patient is unique. If you take 10 cancer patients and you look at their tumors, they might have some main, often called driver mutations that might be shared between these 10 different patients, but the additional mutations that those tumors have might be different. And those mutations might influence both development, progression, and response and resistance to therapies. So
I was just going to say, I'm sorry to interrupt you again. So we've done a number of podcasts with EGFR as one. So would that be an example that if you did some, or is that something different?
Well, actually, EGFR is really one of the poster childs of what is now referred to as precision oncology or precision medicine, because there were at least 2 landmark papers, both of which came out of the Boston area, MGH and Dana-Farber, where they identified patients that were exquisitely responding to an EGFR targeted therapy. And they were interested in understanding why. And because they were using an EGFR, a small molecule inhibitor for EGFR, or a therapy for EGFR, they went and sequenced the EGFR gene, all of those patients, and identified that there were specific mutations in EGFR in those patients that were responding that were absent in the patients that were not responding.
Again, whoever's listening to this should now listen to our podcast on exon. And I'm guessing that's part of what you're talking about. So there are within EGFR, as you say, you think that they would all respond to targeted therapies, but not all of them do. So the exon 20, I believe that was what it was. So we've tried to help people understand that as well. So this is great. It's all.
Yeah, so the way that we're now tackling this problem is by leveraging a whole set of technologies that, again, just like DNA sequencing blow my mind, these technologies blow my mind even more, because these are called genome editing or genome engineering technologies. And they essentially allow a researcher to take cells, any cell type or any organism really, and you can go specifically and introduce any mutation of interest anywhere in the genome. That means that you can rewrite the genome of cells to try to understand how introducing a specific mutation within the background of the genome can influence phenotypes like cancer development.
I think it's incredible. You know, it took us 23 years to read the genome, and we're now at a technology level where we have such accurate pinpoint capabilities to now make edits to it. And that is really a lot of the work that's going on in Francisco's lab. And I am just in absolute awe. It's incredible.
Oh, it's thrilling. I'm so excited. So would one of those technologies be CRISPR? Is that what, because
I think Jordan had a question.
Well, I had a comment and a question. Comment: we've got to get Hildy a lab coat immediately because she's amazing. I'm sitting here watching and listening, and I'm learning from everyone, including you, Hildy. And another comment is just weird in a good way. You said 23 years, and of course I hear 23, and I think 23 and me. I just thought that was interesting.
I never realized that it was 23.
Jane, you can use that.
Chromosomes.
You can use that in your talks to garner big money funding. But I just had one additional sort of generic question, and then I'll let the scientist Hildy get back to the specifics. We hear often, and it sounds like science fiction, that we'll be able to determine at birth or early on who might be likely to be obese later, who might be likely to develop a certain kind of cancer. Is that still science fiction, or is that getting closer and closer? I see Jane, both of you nodding. So Jane, would you want to start, and then Francisco comment?
Yeah, I mean, from understanding and interrogating the genomes, from looking at multiple different populations, looking at the environmental factors that affect these different populations, we are developing something called a polygenic risk score. We've seen a lot of advancement on this, especially in the cardiac field, where we can start to give a score, a prediction of how different people may develop diseases in the future. So that science fiction is already here, and that's already happening. The NIH has a huge project that's driven around this, and that's also how folks like 23andMe do their work as well. They're assessing a risk based on all of the data that we understand from the genome. Francisco, do you want to follow up on that?
Yeah, so I think where the other part that is incredibly exciting is the fact that we can, again, systematically interrogate all these mutations, but which I'll go to in a second. But to just step back, this is not science fiction, it's reality, but there are a lot of ethical considerations that are part of the big debate as to whether, it would make sense to tell a 2-year-old, the parents of a 2-year-old that child might develop a disease later on, because honestly, we don't have sufficient collective information to say with over 97% certainty, for instance, that that's true. It's just we don't have enough experimental information on patient level, population level information.
Yeah, that's a really good comment. And, I can talk personally on a project that I was working on, more population driven and less cancer driven, where we were doing something called newborn screening. So we were doing genetic analysis on newborn babies, but we were very careful and incredibly sensitive to only ever return data that impacted the newborns as a newborn. It is not our place to call out what could potentially be an adult onset disease at 2 weeks old. So there are a lot of ethics around this, and a lot of really careful work that we have to do about how we use this polygenic information.
I was just going to add also, so when I'm not a musician with a jazz group, when I'm not running Upstage Lung Cancer, I am by day also a psychologist. And so PhD from Northwestern, hey. But one of the questions that came up during my internship was the questions, it was around Huntington's chorea. And, at that time it was actually early, a little while back, but it was that kind of question. If you were able to know, if this was in your family, if you were able to know exactly, because some offsprings will get it and some will not. If you were able to know, would you want to know? As a psychologist, that puts you in two different camps. There's some people who would say absolutely, because it will help guide how I'm going to live my life. And other people who would say, that's all I'd be worrying about my whole life. I'd rather not know. So it feels like all of this we're into such an interesting area of psychology and the interface of psychology and medicine, and going forward, and ethics, and errors in prediction. So if somebody said, by the way, when you hit 50, you will have pancreatic cancer, and you live your life in a way that, you know, you're skydiving and everything else once you hit 40, and you don't get cancer at 50. So I think it's just so unbelievably interesting. This is so great. I'm just thinking we should have a second podcast.
Yeah, I was going to talk about a whole lot of other things because we're using these technologies of genome engineering to actually introduce all of the mutations that are seen in patients at once.
Well, this is such an exciting area. I am beyond stimulated and excited by all the information we've discussed today. And I know we have many more things to talk about. So listeners out there, stay tuned. We will have a part 2 of this incredible conversation. I want to thank Jane and Francisco for joining us and sort of sharing this incredible information. Jordan, as always, perfection. And so all of you out there, I hope we've answered many of your questions, or if you've heard terms that you haven't understood, hopefully that's clarified and made genetics a little easier. So thank you so much, and stay tuned. We have more to come.
To find out how you can join Upstage Lung Cancer in raising awareness and funding to beat lung cancer, visit our website, upstagelungcancer.org. We invite you to subscribe and download our podcast, available on all platforms. And we love reviews and ratings. After all, we're showbiz people. There's more entertainment and inspiration to come on the next podcast episode of Backstage at Upstage.